we were talking about the different parts and pieces involved in replicating DNA, right? Remember, I can't see the chat if you chat one. Oh, fish sticks. All right. Oh, okay. Yep, gotcha. Cool. So, one thing that we're going to see a bunch are polymerases. And a polymerase, it's got A's on the end, so it's an enzyme. And polymerases, kind of at the base, basically are able to replicate DNA. So we're talking about prokaryotes, kind of our simplest option with the fewest enzymes to keep track of. So they have 3, finally named polymerase 1, 2, and 3. So DNA polymerase 1 works on the lagging strand. This will make more sense when we actually see what a lagging strand is. To remove primers and replace them with DNA, polymerase 2 is involved in DNA repair processes. So fixing kind of mistakes in the DNA as things are getting copied. And then DNA polymerase 3 will be the one we talk about the most. I don't know why it's number 3, but it is the main DNA replicating enzyme, right? So when we've got a big chromosome, we're making a copy of it. DNA polymerase 3 is the one that is picking up the nucleotides and plugging them onto the DNA, all right? So, come on, everything, there we go. So there are some things that all polymerases have in common, right? They all have, right, and this is that 3' to 5', 5' to 3' sort of thing we've been talking about. All of them have 3' to 5' exonuclease activity. So we mentioned exonucleases. On Wednesday, there are things that can cut nucleotides off the end or the outside, right? Exo means outside. So an exonuclease can kind of trim nucleotides off the ends of DNA. And so what this means, right? So all polymerases are adding things from 5' to 3'. If they can act as an exonuclease from 3' to 5', basically it means they've got a backspace option, right? They can be making DNA and they're like, oh crap, that was wrong. They can back up and cut it off, right? So these polymerases can kind of proofread their own problems to some degree, right? Polymerase 1 has the added ability to have 5' to 3' exonuclease activity, right? So these polymerases are trucking on and they can back up if they need to. Polymerase 1 can kind of jump back and trim things out kind of in the way back, right? And we'll see why in a minute. It's going to be removing primers as we go, which will be important. More words. So many enzymes, But we're just going to knock them all out at once, right? These are the words. So we have helicases, right? Another ACEs word, it's another enzyme, right? DNA is a double helix, really hard to copy something that's all bound up in pieces. So helicase is an enzyme that basically just opens DNA, right? Pulls the 2 strands apart, uncoils it, uses ATP as an energy source to open DNA. And then we have these single strand binding proteins, we'll just call them SSBs. These are we won't talk about these as much. It's just that as we pull that DNA open, SSBs kind of coat it so it doesn't all kind of like twist back together, right? If you've ever pulled like coiled wire or string or something apart, it kind of wants to stay all twisted up. And so SSBs kind of coat the DNA and kind of help it stay open. Now, if you've ever pulled, I don't know why you do this, if you crochet or if you work with rope or something where you've got to pull strands of coiled things apart. If you do that, if you just grab a piece of thread and grab a couple strands and pull it apart, what you'll notice is as you do this and open in the middle, the ends get like extra twisted and they kind of get all knotted up. DNA does the same thing. It acts kind of just like a piece of rope, right? And this problem is called supercoiling, right? So as we uncoil the DNA here, it gets super twisted, super coiled, kind of outside the part that's open, right? So that's kind of a problem. We don't want that to happen. So we have these other enzymes, topoisomerases, that are going to help out, right? So I'm opening the helicases, opening the DNA. Stuff is getting kind of tangled out here. So we have topoisomerases that come in and kind of keep this from getting all twisty, basically. They just kind of come in and help out. DNA gyrase is the one we're going to specifically talk about. Well, not really talk about, just use as kind of our example. So in DNA replication, as we're opening DNA gyrase is over here making sure things don't get super twisty and tangled, right? So it's kind of this situation, right? We've got these things that are copying DNA, and as we open it up, right, the DNA is getting like all messed up out here. So DNA gyrase comes in, clamps on, and just keeps everything nice and tidy. You just keep copying DNA, right? So that's what DNA gyrase does. Keeps things from getting twisty and tangled. All right. So this is the trickiest part about replication. So we're going to kind of walk through it a couple times as we go through this chapter. Replication is semidiscontinuous. What does that mean? So we've talked about polymerases, right? They can only make copies in one direction, right? They can only make a copy in 5' to 3'. Okay. Now, if we remember what DNA looks like, right? It's 2 strands of DNA, right? Actually coiled together into this double helix, right? And they're anti-parallel, right? So this strand is coiled and it's got its 5' end here and its 3' end here. But the strand that's coiled with it has its 3' end here and its 5' end here, which means that if you pull the DNA open and you start copying, right, kind of up this way, one way is really easy because you are adding 5' to 3' and you just kind of go, right? And that's the continuous part. On this side though, you would be adding 3' to 5' and a polymerase, it can't do it. So we've got to kind of do this one in pieces. And we'll look at pictures that'll kind of illustrate this for us. Well, we'll literally illustrate for us because they're pictures, right? So we call these the leading strand and the lagging strand. The leading strand is the one where we're making a copy 5' to 3', polymerase just pops on and does its job. The lagging strand is the one that takes a little more work, right? We're gonna have to pull it open, make a little 5' to 3' copy, pull it open, make a little copy, and then stitch all those copies together, all those pieces. And those pieces that we'll see are called Okazaki fragments because a dude named Okazaki discovered them, right? So, and they are what make up the lagging strand copy of the DNA. Okay, right. So in picture form, how dark does it get if I turn off the light? Right, DNA, right? We're not drawing it coiled because it just makes things messy, right? And it's anti-parallel. So if we have this piece of DNA, one strand is going from 5' to 3', one strand is going from 3' to 5', right? They're going, so here's my 2 5' ends, they go in opposite directions.
So
All right.
Kind of like break down the middle and split off to a different.
So they move off different directions.
Yes, like, yes?
Yes.
Yes. Right, so we're going to start opening this, right? Helicase is going to come in and start opening up the DNA, right? So we end up with this situation, right? The DNA has been opened up. Okay, now, this strand, this is more this 5' to 3' kind of mind game situation. When we open this up, right? This is our template DNA. This is what we're making a copy of. When we open it up, I've got my 5' end here, right? Which means that the end I'm making is 3', which is a problem because I can't make things from 3' to 5'. Down here on the leading strand, right, this was 3', so I'm going to open it up. The matching strand is going to be 5', right? So it's going to be the other way around. It's going to be turned the other direction, which means on the leading strand, I just add a primer here, stick my polymerase on, and it just starts making a copy. And as I open the DNA, it just keeps going, right? So it's continuous. Just as the DNA opens, it's just going to keep copying and copying and copying until we're out of DNA to copy. This guy's a problem because we open it up and the primer needs to be a 5' end. So when I open it up, 5''s in here. So I stick a primer on and the polymerase can make a copy until it hits the end. So then it has to wait until the DNA opens up another tick we add a second primer, and we make a copy until we run into the last primer we made, right? And you just kind of do that stepwise over and over again on the lagging strand. Okay, we'll see it again. Right. Now that opening, right, that we were just looking at, right? We pull the two strands apart. It causes the DNA to have a fork in it, right? It opens up, and it is called the replication fork. It is the opening or the fork in the DNA where replication is happening, right? And this is where DNA primase comes in. Primase is an enzyme that's going to make primers when we're making DNA, right? It's all in the name. So you don't have to worry about this, but it makes RNA primers, right? Because when we talked about, when we kind of initially started about the initiation of copying, your polymerase is great at making copies of DNA, but it kind of needs like a starting point. So we add this little RNA primer that kind of primes the polymerase, right, to attach on and then start copying, right? And then later we'll trim that RNA out because we don't want like little RNA hunks stuck in our DNA, right? RNA doesn't last very long. It kind of breaks down. So we want to go back and replace it with DNA later. But we've got to start with a primer. DNA primer, DNA primase puts it there, right? So this is our RNA primer made by DNA primase. Just put it on the end and start replicating into the replication fork. Yeah, okay. It's not so bad yet. All right, leading strand. We'll talk about leading strand first because it's pretty simple. You set it up and it goes, right? So polymerase 3, right, is what is making a copy of DNA. Cool. But we want to make sure that this enzyme stays attached to the DNA, right? If the polymerase falls off, which sounds kind of silly, but you've got this enzyme working down this super, super long molecule, right? So you want to make sure it stays attached. And so you also have a subunit, right? We talked about enzymes being made of subunits, different pieces. So your cell is going to put the polymerase on the DNA at the primer, and then it's got this little subunit clamp that it's going to put on that's going to hold it to the DNA so that it can then truck on down the DNA molecule, make its copy, not fall off, not do anything funky, right? So you end up with a DNA polymerase with a clamp. And then on the leading strand, right, once you have a primer, polymerase, and clamp, it goes. That's the end of synthesizing the leading strand of DNA, right? There's nothing extra that has to happen there. But on that lagging strand, it starts out the same-ish, right? We open it up, and it has to start at the 5' end, right? So we've got to put the 5' primer in the replication fork, basically. But it works the same way, right? You put the primer on, put the polymerase on, you put its little clamp on, and it synthesizes a piece of DNA. Just the same, right? DNA polymerase 1 is going to come in, pull that primer out, right? We're going to add a new primer further into the fork, right? Because the DNA is kept opening while we were busy, right? And then synthesize a new piece of DNA and crawl it forward and do it again, right? Now, we're putting these primers, though, kind of periodically through our DNA, and we don't want them there. Fortunately, polymerase 1 is going to go through and pluck them out, replace them with DNA. But now we've got pieces of DNA that aren't connected to each other. All these little Okizaki fragments, right, are just kind of stuck here. And we want the DNA molecule to be like a whole molecule at the end. That's kind of part of the point. So we've got one more little enzyme that's going to come in, DNA ligase. If you know the word like ligature or to ligate something, right, it's going to come in and stitch all the pieces together, right? Ligatures, like stitches. It's going to come in and attach everything together, right? Yes?
Do we need to know DNA pull 3 and DNA pull 1? Do we need to know specifically? Or is
I'm
it-
actually not going to have you know, yeah, pull 1, pull 2, pull 3. I mean, I'll use the words, but I'm not going to have to make you, right? I mean, I will say polymerase 1 in a question, but you won't have to actually remember which is which. Does that?
Okay.
Yeah.
So it won't come up as like.
I'll be like,
so-
which polymerase? No, it'll be, yeah.
Okay. Just making sure. 'Cause that was-
Yeah. I mean, polymerase 3 is the one that does the making a copy. That's kind of, if there's one that's important that you want to remember, polymerase 3 makes a copy. But other than that, I mean, I don't, nah is the basic answer. Right. So again, here's what's happening on that lagging strand, right? We opened it up a little bit. We put in a primer. we made a copy, right? We opened it up a little bit more, we put in a primer, we made a copy. All right, this primer is still here. So DNA polymerase 1 is going to come in, chop out the primer, and replace it with DNA, right? DNA ligase is going to come in and stitch this together. And then same thing, primase adds a primer, we make a copy, we chop the RNA out, replace it with DNA, and stitch it all together, right, until we run out of DNA to copy, right? Right, and that's how you copy DNA, right? Oh. Here we go. All right, so copying DNA has 3 stages. Initiation, right, starting it, opening the DNA and adding the primers. It has elongation, stage 2, copying the DNA. Stage 3, termination, right? We're talking about prokaryotes with one little circular piece of DNA, which has an origin site and a termination site. So termination in prokaryotes happens at the termination site, right? We make our copy from the beginning to the end. When we hit the end, DNA gyrase, the thing that was helping us keep everything nice and tidy, pops both the pieces apart. We've got 2 new chromosomes, right? Or we've got 2 copies of the same chromosome. And that is the end of DNA replication. Okay. It's manageable. Okay. Now, all of these pieces, right? Helicase and primase and polymerase and gyrase and a clamp loader, which we'll see later, but we're not super worried about. It's just going to show up. All of these different enzymes and pieces and proteins and things that are all copying the DNA. We collectively refer to this whole mess of stuff as the replisome, right? It's like a whole engine made of a bunch of different parts. And so it gets kind of this collective name, the replisome, that does DNA replication. So it's kind of like, you can think of it as the replication organelle. It's kind of a temporary organelle. It gets put together, it does its job, and then it gets kind of taken apart again. But so the replisome. So when we want to just talk about everything that's doing the DNA replication, we can just call it the replisome and not worry about listing out a dozen different enzymes. Again, 2 main components, one being that primosome, right? Open the DNA, put the primers on, coat the DNA, right? And then those 2 DNA polymerase 3s that are actually making the copy, right? So all those pieces together are the replisome. So when you see that labeled kind of in pictures and things, that's what it is talking about. All right. A more detailed version of the replication fork. So it's kind of a crazy picture to look at first, but what it is, this is a picture with all of the, like it's all of the pieces in the same picture. All right, so if you want to kind of like pick through this, right? So here's helicase, right? We'll kind of go in order of guys we've introduced. Helicase is working through here and unzipping, right? Opening up the DNA. DNA gyrase is working ahead to make things not all twisty and tangled, right? So we're opening the DNA, we're making sure it stays nice and tidy. Next, we have primase. And so primase on the leading strand added a primer out here, and now this DNA is just being copied, right? Just straight on till we hit the end. On this lagging strand, it's having to add a primer, and then we're making a copy, adding a primer, making a copy over and over again. All right, we'll walk through this kind of in a series of steps. This image adds in this clamp loader. Don't worry too much about the clamp loader. It's just a piece here that is holding the polymerase onto the DNA and putting these clamps on that holds the polymerase onto the DNA. Okay, so we've got all of these. None of these are new words except the clamp loader, and he's a weirdo, so that's fine. Nah. He's just here doing a job. All right, so DNA synthesis at the replisome, right? So this is the replisome. All of these pieces together are the replisome. So step 1, right? DNA polymerase 3, guys making the copies are active on each strand, right? We've got 2 polymerase 3s going on, right? On the leading strand, primase added a primer back here. The clamp loader, pop polymerase 3 and a clamp onto it, and it's going, right? So we don't have to worry about the leading strand anymore. It's doing its thing, right? On the lagging strand, we opened the DNA up a bit, primase put a primer, the clamp loader put polymerase 3 here, right? Because it's got to copy this direction. It can't go that way. So we've got to put a primer and make a copy. Well, now we've run out of space. So we open it up some more, we put a primer, do it again. Take the polymerase 3, little clamp, attach it, and make a copy, right? And while it's going this direction, helicase is opening up more DNA, right? So polymerase is here copying, primase adds a primer, right? Kind of at the beginning of this little open uncopied loop. And then this whole process just kind of keeps moving to less. The primase added a primer, it drops off, we don't need it now. The clamp loader, once this copy is done, pulls polymerase off and drops the clamp. It's going to pull polymerase 3 back to the next primer and put on a new clamp. Polymerase 1 is going to come over here, pop this primer out and replace it, right? And then ligase is going to stitch all that together. So we get this situation, right? Clamp loader comes over here, slides the polymerase in, puts the clamp on, and it starts making this copy, right? Polymerase 1 has popped that old primer out, ligase kind of stitches it all back together, right? And this just happens over and over again, all the way down the DNA. Right? And that's just kind of a picture. It keeps going, right? Keeps going. So now this guy's making copy here. Helicase is opening this more. Primase is going to come in, put another primer, and we'll just do it again. And again, and again, and again, and again, until we're out of DNA. All right. So that was prokaryotic replication. We're not going to walk through eukaryotic replication. We're just going to kind of point out a couple things that make it different. And that'll be enough for us. So we're going to kind of compare the two, talk about what is structurally different and why we have to care. So, as with kind of all of these things, right? Cell division was more complicated. DNA replication is going to be more complicated, all right? For kind of obvious reasons, right? We were talking about bacteria. We've just got a little circular single chromosome, make a copy, and you're done, right? One and done. Eukaryotes have much, much, much more DNA for starters, right? We have a lot of DNA. It's on multiple dozens, sometimes like hundreds of chromosomes, right? That we have to copy and keep organized. And those chromosomes are condensed down and wound up and wrapped around proteins, right? It's a really messy kind of molecule. And so your cell has to have a lot of other things in place to make sure things stay organized and end up where they need to be, right? Organization, it's the name of the nucleus game. So, got more DNA, it's on multiple chromosomes, and your chromosomes are linear, which doesn't initially seem like it would be a problem, but we're gonna see where it's gonna be a problem. So, basically, same enzymes. They get different names. We're not going to worry about their different names, but you're going to have the same process, right? You're going to have polymerases that are making copies. You're going to have helicase that's opening DNA. You're going to have gyrase that's untangling stuff. But what's going to happen is you're going to have multiple replicons. All right, we said the word replicon on Wednesday. Bacterial chromosomes, single circles, have one point of origin, right? And they kind of just make their copy in one go. That is a single replicon, right? Like kind of a single replicated unit. We have so much DNA that if our chromosomes each only had one little origin point, right? If we just started at the beginning of every chromosome and copied it to the end, it would just take way too long to copy all your DNA. You just have way, way, way too much. So each of your chromosomes actually has kind of multiple origin points. So it's made of multiple replicons, right? So that when we want to make a copy, we can kind of open your chromosome up at a bunch of different places and then stitch together a bunch of copies. It's just more efficient, it's quicker, right? It's not super specific, it kind of varies across your life. So early in life, when you're developing and growing and you've got to divide cells like super, super fast, you have more of these replicons, right? Your DNA is being replicated like super duper fast. As you get older, right, you're not growing theoretically as much. And so you can get a bit, you don't have to be as rushed, right, in cell division and DNA copying. Okay. So the way this happens, right, because we don't do binary fission, right? We have to go through mitosis or meiosis. And so S phase, right, that's DNA synthesis phase. That's when all of this happens. But your cells actually kind of prep in the interphase a little bit. So what they're going to do before S phase is they're going to go ahead and pop all those helicases in, right? So everywhere your body wants to start making DNA copies, it pops a little helicase in there so that when we get to S phase, we can start opening the DNA up, right? So all the little helicases get loaded onto different origins across all the chromosomes. And then in S phase, the helicases start to turn on the replisome, right? All of those enzymes come together, start to assemble, and then it's S phase, and you start copying DNA. And mechanically, it looks a lot like, at that point, at each replisome, it looks very much like the process we talked about with prokaryotes, right? Now, you don't need to know these two names, but priming uses a couple of polymerases. They get a different name. It's called polymerase epsilon and polymerase delta. That's fine. We don't really care for our purposes. But instead of having, right, in prokaryotes, you had two polymerase 3s on the leading strand and the lagging strand. Here we just give them different names, polymerase epsilon on the leading strand, polymerase delta on the lagging strand. We're not too worried about that. All right, so we can deal with our having a lot of DNA and having big chromosomes, right? We can make lots of replisomes, we can do lots of copying at the same time. The other problem is our DNA is linear, which seems honestly like it shouldn't be a problem, except we're gonna have to play this 3', 5' game again, because it's gonna cause us trouble. They have ends. And so one thing is, your body has to protect the ends of your DNA, right? So we've got these exonucleases, these little enzymes like flipping around, and they will cut nucleotides off the ends of things, right? Because enzymes are good at what they do, but they're not very smart, right? When enzymes bump into a molecule that they can do something to, they're just going to do it, right? So to protect the ends of your chromosomes from nucleases, you have these kind of long, repetitive, specific sequences. They don't code for genes. They don't get used for anything, but they're kind of a buffer, right? So something can happen to the end of the DNA, and the DNA you need, the stuff that is coding for the things that you need, is going to be fine, right? So it maintains the integrity of linear chromosomes, right? And these telomeres, right, is the name for these little chunks of sequence on the ends of your chromosomes, are not made by the replication complex. The replisome doesn't make these copies because, right? It's this lagging strand again. It always causes trouble, right? So over here, right? Leading strand is just copying until we get to the end, we're done. But on this lagging strand, as we open it, we put a primer and we copy, we open it, we put a primer and we copy, we open it, primer, we copy, we open it, we're at the very end. So we put a primer on the very end, on the 3' end of the lagging strand. We can't do anything. Our polymerases don't work on the 3' end. So we cannot replace the primer with DNA, but you don't want to leave RNA out there because it's going to fall apart and degrade and not be any good. Right? So you end up with this problem, right? So here's my lagging strand, right? Primer Okazaki fragment, primer Okazaki fragment. We're doing great. We replace everything, but now I can't fix this last primer. It gets pulled off with RNA. It's no good to us. But now one of my copies is too short. So the next time this chromosome gets copied, we just make a copy of the short copy, except for the primer that we lose. So then we make a copy of the short copy, right? It's going to keep, your DNA will just vanish over time, right? So your body has to figure out some other way to deal with being linear. It's a problem. So we've got a bonus enzyme to deal with our linearity, right? Telomerase. Easy enough. ACE means it's an enzyme. It's a telomere enzyme, right? And so its specific job is to synthesize the telomere repeat sequence. And it's kind of cool. It's like this little kind of clampy tube-shaped guy. And he has his own, basically he's got his own primer inside of him. So if he attaches to something, he can kind of like prime his own reaction. Here, look at the picture. Right? Here was our problem. I've got these bases hanging off where the primer's gone, right, on my lagging strand. This guy, telomerase, inside of him has a matching sequence, so he can attach here. And then he's got this little kind of extra primer where he can attach a few more bases. Okie. Now, he can, and it's repeating, so he can add these bases and then slide himself down to the end, right? Because it's repeating. He matches here and he matches here again. And so now he's like a little dummy primer. Now, we can come in and synthesize this last little piece, and he's just going to break off these little nucleotides that he added, right? They weren't part of it to begin with. It doesn't matter, right? So we can take that with us. And now we have fixed our lagging strand problem, right? So telomerase is used in organisms with linear DNA to sequence that last little piece of the lagging strand. Okay, that's not so bad. Now, as with everything, we've got to regulate telomerase because we don't need it to work as hard all the time, right? When you're young and you're growing and you're making loads and loads of cells, you need telomerase to like be on it. So your telomerase is really high functioning during development, during childhood. But as you get older, right, your somatic adult cells that tend to kind of hang around for longer, telomerase kind of like, I don't know, takes a break, right? You've got some cells that are going to keep dividing quicker. Telomerase is going to keep working in those, but it starts to get a bit lazy. And this is what leads to aging, right? The ends of your DNA, as you get older, basically your DNA gets a little raggedy. Things start to get a little lazy, a little bit more haphazard. Your DNA loses some elasticity. This is where your skin starts to get a little loose. Things start to get a little funky, right? Because the copying gets lazy. And when the copying gets lazy, the product is not ideal, right? Now, it always is working enough. If you have no telomerase, you basically just do not function, right? They remove telomerase for some mice. After 6 generations, there are no more mice. You can't, right? Eventually, you have just wrung off all the ends of your DNA and things just are going to stop working. Conversely, telomerase can get kind of ticked up in cancerous cells, right? So cells that you would want to senesce, that you would want to kill off to not function. One of the things that will happen in cancer cells is you'll see an activation of telomerase that will maintain the cancerous DNA, which is unfortunate. It's kind of the opposite of what you would want, right? So they end up maintaining their telomeres kind of at a higher level in cancer cells than like the rest of your kind of chill somatic cells, right? All right, which I think gets us to the last chunk. So we're making copies. We're taking care of our copies. We're doing okay. But we're still going to have to fix stuff. We're still going to make mistakes. DNA is still going to get damaged, and we need to have our DNA not full of mistakes and not damaged, right? Otherwise, it doesn't work. So damage to your DNA is happening all the time. Some of it is just errors due to replication, right? But we've got our DNA polymerases that can do a little proofreading, right? They've got a little backspace button. They're like, oh, I screwed that up. We'll go back and fix it, right? So that's happening at one level. But we've also got this mutagen problem, right? You're exposed to chemicals, to the sun, to radiation, to all kinds of stuff that causes your DNA to change faster than it's supposed to, right? DNA kind of mutates over time. That's natural. It should be a slow and easy process, but we get exposed to things that causes our DNA to be damaged, basically. Right, so we've got, there are lots of ways. There are lots of ways that your body handles this, because it's really important. It's really important that you've got good quality DNA that is coding for the things that you need. We're going to talk like about 3 of them. Right. So let's do it. We can lump all of them basically into 2 categories. You have DNA repair that is specific, right? You've got a specific sort of problem, and we're going to fix right away like a very specific tool that fixes a specific problem, right? So we can have specific repair, And then we can have non-specific repair, where you've got this single mechanism that can just come in and fix things in the DNA no matter what the actual problem is. We'll look at examples of both of these. So repair can be specific or non-specific. So mismatch repair is kind of a non-specific, it's just kind of a general thing, right? And this is that kind of like that polymerase proofreading thing, right? So if you have a mismatch base, there's stuff in your cells that can go in, remove the incorrect base, put the right base in, right? By copying the template strand, right? This is what those DNA polymerases are doing when there's a mistake in DNA replication, right? So mismatch repair, mismatch base, pull it off, put the right one in, and you're done. A little more complex, an example of specific repair is photo repair. And this is one that probably has to happen a good bit, right? So we know about skin cancer, we know about skin damage from the sun. One of the things that is happening, one of the problems is when you are in UV light, the energy from UV light, if you have, and you do, everybody has places in their DNA where there's two thymines next to each other in the sequence. That's a completely normal thing to happen. UV light reacts with these thymines, breaks them loose of their base pairs and clicks them together and causes what we call thymine dimers. So it kind of breaks and puts a little hitch in your DNA, and that's bad. You don't want busted DNA. So you've got a specific enzyme whose only job it is to fix thymine dimers caused by exposure to UV light specifically, right? It's a very specific problem, but it's very common, right? We're exposed to UV radiation basically all the time, right? Kind of funnily, photolysis is powered by visible light. So it uses power from visible light to fix a problem from being exposed to light, ironically. But either way, photolysis comes in, pops the thymine, right, cleaves the thymine dimer and puts it back in place. Right? Fixes the problem. We've got kind of a specific repair option. Another non-specific option is excision repair, right? And all this is you have enzymes that when there's some mistake, right, some damage or mismatch base, you have enzymes that will come in and cut out, excise the damaged spot, right? So we recognize damage, we just cut the region out altogether, and then we've got these DNA polymerases floating around and they're like, oh man, unmatched DNA, I'm on it. It comes in and fills it in, right? So we have excision repair, where the damage is just removed and another enzyme will come in and fix, right? Because you've got the template strand, right? You've got this second strand, so you know what goes here. You can just replace it with the bases that it needs. All right. So we've got mismatch repair, photolysis, right? Photo repair, and then this excision repair option. And there are loads of others. We're not going to worry about them. All right. And that is the end of chapter 14. All right. So we've got, not too bad, a few minutes to talk about the exam, maybe do a little bit of chapter 14 review. So the exam is going to look format-wise real similar to the last one. It's about 30 multiple choice, handful of fill in the blanks with a word bank, and a bit of matching. You guys have been letting me down on the matching. You gotta come through on this one. And like maybe a couple little short answer-y kind of things. Punnett squares, know how to do Punnett squares. Hint, hard. All right. But structurally, it's gonna look like the other exams. All right. I put a load of review questions on this one. We're gonna walk through, let's pull a couple of them out so we have time to. Maybe go back to the beginning since it's been a little while. So just the structure of DNA. Structure of DNA. So DNA is a giant polymer made up of monomers. What are the monomers of DNA? A little bigger than that. What are the pieces that we're going to link together to make a big DNA molecule?
Sugar.
Bigger than that. I mean, we can start there. What are the three pieces of that monomer? The sugar, 5-carbon sugar, a phosphate group, and yep, a nitrogenous base. So when all those guys are linked together, sugar, phosphate, nitrogenous base, that is a what? Do you know what that is? close. It's a nucleotide, right? So a big long chain of nucleotides then is a nucleic acid, right? And so we make these long chains, right? Linking sugar to phosphate, sugar to phosphate, sugar to phosphate. What do we call that thing, right? The bases we're not worried about yet, but that long chain of sugar and phosphates that kind of make the backbone, as it were, of the molecule. It's called the what? Starts with P, ends with backbone. pH.
Phospho.
Phospho.
Lipid.
Lipid.
Phosphodiester.
Yeah, phosphodiester backbone. Right, so you got a phosphodiester backbone with a bunch of nitrogenous bases attached. And so a full DNA molecule, right, is two of those twisted together, right? We've got our full double helix DNA. All right, what else? So we say that DNA replication is semi-conservative. What do we mean?
You said it's a full twisted DNA molecule.
So the two strands, right? You've got phosphodiester backbone with bases, complementary strand twist together, and that's your full double helix, complete DNA molecule. Yeah. What do we mean when we say that DNA replication is semi-conservative? I have a parent strand of DNA that is two, right? It's two strands. My parent DNA chromosome is two strands wound together. When I make a copy, so then I have two DNA molecules. Is one of them made of two parent strands and one of them made of two daughter strands?
Daughter strand each consists of a parental strand and one strand.
Right. So in each new copy, you have conserved one of the parent strands and added a daughter strand. So it's semi-conservative. Okay. 3 stages of DNA replication.
Stage 1 is imitation. Replication begins.
Right. Replication begins, initiation.
Initiation.
Uh-huh.
And then elongation.
Elongation, yeah. And then finally, termination. Fab. What are DNA polymerases?
Matches exist DNA bases that complement nucleotides and stuff.
Yeah, they can actually make new DNA strands. That is their job. All right. What are some things that DNA polymerases have in common?
They add new bases to 3-inch strand of existing strands and synthesize in 5 to 3-inch direction. Requires a primer of RNA.
Yeah, yeah. So they can add bases to the 3' end, which means they can synthesize DNA from 5' to 3', right? But they need a primer. We've got to have a place for them to start. They can't kind of start on their own. All right. What is supercoiling? That was today.
Yeah.
Right? That is, there we go. Right, supercoiling. So as you open, right, if you take a piece of rope and pull it open in the middle, the ends become super, super twisted. That's called supercoiling. How do we prevent supercoiling? What prevents supercoiling? Topoisomerases, the one example we gave was which one? It was an enzyme, which one? DNA gyrase, absolutely. All right. For the sake of time, we will not walk all the way through DNA replication again. Why is eukaryotic DNA replication more complicated?
Multiple replications involves multiple.
Yep, we have multiple origins. Why? Why do we have so many replicons?
Because there's errors due to replication.
Back up. What's different about us, our DNA?
Complex packaging.
Right, we've got really complicated, we have lots of DNA. It's on lots of chromosomes. It's packed up really complicatedly, and our DNA is complicated. But it is also different from bacteria and prokaryotes in that it is, what was that one other thing about our DNA?
I didn't write that down. I'm about to.
Prokaryotic DNA, they have one chromosome, and that chromosome is a
Non-existent. First letter.
it's a circle. Is our DNA in circles? No, it is.
Double helix structure.
I mean, well, so is a prokaryotic DNA. Not a circle. It's
A rectangle.
a line. It's linear. It's linear. Yeah.
Oh, yeah, I said linear structure.
Did you?
Yes.
Oh, sorry.
That's crazy.
It got lost in there. Okay. Anyway, keep reviewing, keep working. Don't forget the Unit 4 assignment. If you have any questions, come find me, email me, all those sorts of things.